Good evening, everyone. Good evening, colleagues, students, friends, and family of Prof. Keyster. We will soon be starting with our program. My name is Professor Admaya Dube, and I'm the Deputy Dean for Learning and Teaching, and I'll be the Program Director for this evening. We'll soon start, and we'll have an academic procession. So I will ask everyone to rise for the academic procession, and also to remain standing for the national anthem, and then we can be seated. But I'll give you further guidance. And then also just one change on our program, that's on point number 7, which is a question and answer session. So we will no longer have a question and answer session. We will have some discourse as part of the responder responding to the inaugural lecture. So that's the only change on our program. So we can remain waiting and wait for the procession. Okay. So you may all rise for the academic procession, please. Anthem will be led by Ms. Simamkele Tempi, soloist of the UWC Creative Arts Choir. Please be seated. I now call upon Professor Tyrone Pretorius, Rector and Vice-Chancellor of UWC, to welcome you all.
Good evening, colleagues, students, and distinguished guests. It is a great pleasure to welcome you to this special occasion, the inaugural lecture of Professor Marshall Keyster. I extend a warm welcome to the deans and the deputy deans of faculties, and of course to our inaugural lecturer, Professor Keyster. We are also delighted to welcome Professor Keyster's family, friends, colleagues, and students who are joining us here in person tonight. It is wonderful to have you all here to celebrate this occasion with the University of the Western Cape. Inaugural lectures are special events in the academic life of a university, and it is to be seen as a significant milestone in the career of our academic staff. Whether one is appointed to the rank of full professor or you achieve that rank through promotion, the path is a very arduous one. It's a journey that warrants a special place in our academic calendar. I'm personally pleased that we have taken the decision to resume with these inaugural lectures. The last one I recall was in 2016 and was disrupted by Fees Must Fall. This event, the processes and the protocols associated with it, has long left our memory. And we have, especially the registrar and I, have amicable debates about the correct protocols and the correct processes. Hopefully, Professor Keyster's inaugural lecture tonight relaunches this prestigious event in the academic life of the university. We have a number of inaugural lectures that will follow for the remainder of the year. And if I'm not mistaken, we have one from each faculty. This evening, we celebrate a remarkable individual and his groundbreaking work in the area of plant biotechnology. Professor Keyster's lecture, titled Decoding the Plant-Human Health Nexus for the Greater Food, promises to enlighten us on the vital role plants play in our lives. I would like to extend our gratitude to everyone who has contributed to making this event possible, particularly the organizers, the then acting Dean of the Faculty of Natural Sciences, and the current Dean of the Faculty of Natural Sciences for their support. And of course, a great big thanks to the Registrar and her office for tending to the logistics. With these few words of welcome, I would now like to invite Professor David Holgate to introduce our inaugural lecturer. Thank you.
Thank you, Vice Chancellor, and it's lovely to be here, colleagues. Just to follow on from what Professor Pretorius just said, the fact that this is the first inaugural lecture in a long time brings us great joy, and of course the fact that science is leading the way yet again is of course something that brings us great joy too. So it's my honor to introduce the lecture today. So Professor Marshall Keyster fits the theme quite well, because we often talk about growing our own timber. I guess we could describe Marshall as a sturdy UWC oak. Marshall matriculated in Belleville South. He started his BSc studies at UWC back in 2002. And after completing his BSc degree in minimum time, Madam Registrar, he pursued his BSc honors, followed by an MSc degree in molecular biology at the UWC Institute for Microbial Biotechnology and Metagenomics. Marla is here somewhere, our current director. He then proceeded to PhD studies in plant biotechnology at Stellenbosch University. And after graduating in 2011, he returned to UWC, first to pursue a postdoctoral position before becoming a member of the academic staff as a lecturer in biotechnology in June 2012. So Marshall's career has grown firmly and steadily at UWC since then. His progression through the ranks of senior lecturer to associate professor and finally full professor in 2022 has been accompanied by an establishment of his research presence, which was witnessed by first a Y2 rating early in his career and most recently a C2 rating from the NRF. For those who are not academic colleagues, it sounds like a C is a bit like 60%. But if you have a C rating, that's a firm recognition of the standing of your research. So it's no small accolade. Marshall also served as head of biotechnology department along the way. So an academic establishes his standing not simply locally, but also amongst international peers. And Marshall's CV shows his rootedness at UWC, but with an international reach and reputation of which we as a faculty are very proud. He's an associate editor of the journal Plant Molecular Biology Reporter. He's got 60 research publications, 11 peer-reviewed conference proceedings, as well as 4 book chapters to his name. He's one of 40 mentors in the Food Systems Network for Africa Fellowship Program. One significant milestone in Marshall's career was the award of a Missouri University South Africa Exchange Program grant back in 2015, and he spent 3 months at Missouri at that time. And that was significant for Marshall as well as his colleagues and students, as it's fed a lot into the program of research at UWC. He's raised over 10 million in grant funding since 2012, yet he can't even afford a barber. Currently, Marshall is serving as a reviewer for over 20 plant science journals. He's hosted 6 postdoc research fellows, graduated 87 postgraduate students from honors through to PhD level. 4 of his PhD students are working as academics at local and international universities. Marshall has examined many dissertations, served on NRF panels, been an NRF reviewer for research grants and scholarships and rating applications. He's attended and presented his work at conferences across Africa and abroad. He's a member of 5 scientific societies, including the prestigious International Society for Experimental Biology, and he's currently supervising 28 students. So his research aim is to understand how soil contaminants, mostly heavy metals, specific to South Africa, which are often produced by mining, how they impact food and animal feed crops. And because heavy metals are also a human health risk, he's expanded his research into the One Health theme in order to decode the plant-human health nexus. He does have time for family, despite all this work. Marshall was born and raised in Greenlands, just around the corner, to his parents, Adam and Maria Keyster. He's the youngest of 8 living children and is married to Jessica Keyster, and they've got 2 children, Mary Jane and Adam. Marshall tells me that he's been drawing since he was a young child. It's something I didn't know. It's become an integral part of his life. He also loves photography, capturing moments and emotions through the lens of a camera. And he's a big supporter of the national rugby team. Of course, one of the disadvantages of being homegrown is that your previous lecturers are now your colleagues, and they probably remember many stories of you as a student. One that Marshall recounted was Gavin Marnefeld gave him a 0 and a smiley face on one of his assignments because he didn't use proper references to back up his hypothesis in second year. But those of us who know Gavin's a great educator, so he did give Marshall a second opportunity. The other stories that he tells are of how he commuted to Stellenbosch when he started his PhD studies, and it speaks to the realities of our students, missing the train on occasion because he was standing at the wrong platform, but also sitting in a taxi and receiving a phone call. Usually one's quite nervous to take your phone out your pocket when you're in the taxi. And then he thought it was a prank call from the NRF because it was in April, but they called him to say that he'd got his NRF bursary. I don't know how many of our students have heard on a taxi that they've got an NRF PhD bursary. So Marshall's an all-rounded human being. He's a wonderful colleague. We're very proud of you in the faculty, Marshall. And as I said at the beginning, you're a significant UWC Oak. And it's really appropriate that you're going to address us with your inaugural lecture, Decoding the Plant-Human Health Nexus for the Greater Food. Thank you.
Good evening, ladies and gentlemen. I would like to start off by saying all protocols observed. So, as I was reflecting on this lecture, I must say it felt like this is my final lecture at UWC. I don't know who was going to go through the same process, but that final moment before you walk after the rector, that is a terrifying walk. So I would like to start off by saying thank you for being here, and those who's online as well. So why are we studying plants? While I'm standing here in the growth room, I'm exhaling, and that is actually a vital cycle that keeps us alive. So I'm giving the plants carbon dioxide, and the plant is giving me oxygen. So I don't have to explain further why we need to study plants, because this plant is life. But also when I started my studies, I realized that we are giving plants what they need to survive, and they are giving us the necessary elements to survive. And what is interesting is that those elements overlap. And by studying this interaction between us and plants gives us an opportunity to work on the Sustainable Development Goals of the UN, United Nations, which touches on zero hunger, as well as good health and well-being. So my talk today is 100 slides, don't worry. But I'll start off by briefly describing what I did in the past, and then moving on to what I'm doing now, and then moving on to the future. That is not me, by the way, and it's not going to be me in the future. So before I start, I would like to say a dedication to the two people that really stood behind me and got me here. So my mother, Maria, is on this side, and my father. And one day when I'm big, I want to dress like him. And also, I had to crop out my brother, because this was his graduation. And at the back is the student center. So we have always been UWC people. Okay, so just where I come from. So this is our house. And back in the day, it used to be called Bosbo. And it was part of with what now is Cape Nature. And it was only 30 houses. And where the houses is now in Greenland, that used to be bush. And so there is my connection with plants already. This is not, this is the road actually. And it's not tarred, as you can see. So yeah, humble beginnings. And I asked the Meta AI to recreate a moment when I decided that science would be my career going forward. But you can see Meta is making the rain inside the house. So I remember the story where my mom told me about the water cycle. And never did I know that or realize that this cycle will come back in later years when I'm now a professor studying plants. Because she used to tell me that the water that comes back is vital for plant life. So I don't know if you can see, but so this road here is where I come from, and this is the boundary of UWC. So it used to take me 10 minutes, and on days when you have to run for sketchy characters, it will take me 5 minutes. Okay, so then so the first part of my journey is when I met Prof. Don Cowan. And I don't know what he's calling here, or if he's just posing for the photo, but he used to have RChem and IBM. He used to be the director. And so Don took me in and basically raised me that first few years. So he was interested in all of the microbial life in the Antarctica, in Antarctic region. And one of the groups of microbes was cyanobacteria. And I remember studying cyanobacteria when I was in my undergrad. So why are they important? They are just similar to plants. They are also very important for life. They are also responsible for giving us oxygen, carbon and nitrogen fixation in the soils. And so they are actually the farmers of the old, not the modern, but the old age farmers. So that is me in my MSc. I still look the same. And the gel on this side, it looks very interesting, but the reason why it's there is because I clearly remember struggling to get this gel. And it was because I couldn't get this lane here, which is the negative control. I couldn't get it clear. And all of the molecular scientists will tell you that is a scary thought. For a full year, I was struggling. And I remember in my second year, on a Saturday, I came in, and something told me I put the gel on, and I didn't really pay attention. And then I took that gel because I expected something to be in this lane, something that looks like this. And I chucked the gel in the bin. And then something told me, just look, this is it. And then when I put it back, it was actually clear. And then I graduated my MSc. So I was looking at cyanobacterial diversity on the slopes there in Antarctica. Unfortunately, I never got to visit there. Some people tell me it's a very, very beautiful place, but yeah. So I did get cyanobacteria at the bottom of the slope, but at the top, not so much. Interestingly, I got this group called Acidobacteria. And now, in later years, we managed to get a big grant. I'm not going to say how much. We got a big grant to work now on Acidobacteria. And I'm telling the people in the research group that I ran away from ecology, and now they're bringing me back to ecology. Okay, then I drove with my yellow Mark I down Merriman Street, and I landed in that building where I met a young gentleman called Professor Ndiko. And I remember the first day when I started, it was in 2008, Prof. Ludidi gave me a tube and said, go and work on this. This is your whole PhD. And he was actually right. So this was my first meeting now with plants. And this is me as a PhD student. and I still look the same. And during that time, I established myself very quickly as the biochemist in the group. I'm not going to say before who arrived, but yeah. So this used to be my life, setting up these essays and then reading it in this spectrophotometer. And what I studied was the interaction of nitric oxide as a signaling molecule in maize as well as soybean. That was my PhD. And what we knew back then was that nitric oxide regulated everything inside the plants. So you can see here, seed germination, root development, flowering, senescence, mineral nutrition, that's just the uptake of nutrients. And up till today, no one can find the nitric oxide synthase enzyme. And yeah, it's an amazing thing that we all know that the plants make nitric oxide, but we can't find the enzyme. So this was my first publication. And I worked with Jacobus Leeds, who's this gentleman here, who was very good in growing plants, but not so much in biochemistry. So, and I was responsible for doing most of the biochemistry, and he was responsible for sorting out the plants. And what we showed during that time was that these bumps on the roots here is called nodules. And inside these bumps, you have microorganisms, bacteria, which is very important and which allows the plants to make nitrogen, or the bacteria makes nitrogen, and it fixes it inside the nodules. So the plants doesn't have to get so much nitrogen from farmers. So farmers will coat the roots with the bacteria and then grow the plants. So, and these were the findings. So when we treated with the inhibitor, we saw that we don't get nodules. And so we had to redo the experiment and first get nodules and then treated with the inhibitor, and then saw that when we do that, we get a whole range of activities being inhibited, like nitrogenase activity, which is responsible for nitrogen and cell viability. But when we added a donor of nitric oxide, something that gives nitric oxide, we reversed these effects. And that led us to the hypothesis that nitric oxide resulting from nitric oxide synthase activity is required for development of functional nodules. Then I was also busy with my own work while I was helping others, and I was struggling again for a whole year. So during this time, the second gel was now standing in front of my door, which is this one here. And I couldn't get these bands here and here to come up on the gel. And later we discovered that somebody mislabeled the tube. And that was all it took. And then I did this experiment where I showed that when I added the nodule extract, which have now which I combined with the protein, then I basically decreased cysteine protease activity. And the same when I used the pure protein. So at this point, I would just like to acknowledge the 8. I'm not going to keep the photo on too long because, yeah, people are going to feel all kinds of ways. I want to thank my nieces and nephews, all my school, uni friends, my teachers, and also the in-laws who's now married to my brothers and sisters. Then, during that time, the people in IMBM, there's others also here. There were, Mala will recall, 20 or 19 postdocs. I can't put all of them here. Strangely, Mala was also one of them. But so this is the people that worked directly with me, Anna during my honors, Lucas during my MSc, and then my PhD in Deco's group, which were called the No Group. And then 2 gentlemen from Hungary, which assisted me during that time. So, and also Zainu. Zainu Arif, yeah, she also took me up for an honors. So I must mention Zainu Arif. Okay, so then I came back to Greenland, maybe against my will. And I established, started my lab. It looks still the same. It's still messy. And I would like to just remind everyone here that knows biotech or is touched by biotech that we are in biotech to develop and create products, process technologies for various applications. This can be medicine, medical. It involves using biological knowledge, and we manipulate, and we change, we edit, and we do this to develop new products. And this is basically what I'm doing. So for my family, who thought I was just a normal, random guy who walks to campus, this is what I've been doing. And biotech is extremely broad. The biotechnologists will tell you. Okay, so the plant-human health nexus is the next batch of work. And here we study how does plants and plant-derived compounds impact human well-being. This is health, nutrition, and also anything that's related to that. If you eat plants, there will be some changes in your gut. This will make you feel happy, or it will make you feel sad. But yeah, that interaction is the plant-human health nexus. And this is nicely illustrated in this publication. So we published an article where we came to the conclusion that some biomolecules which plants make can be used to overcome certain stresses. And this is something that we started off when I came to UWC. And these are some of the molecules that when I came back here, which I investigated. So I moved away from nitric oxide, and I moved into studying these molecules. And all of the molecules are made by plants. So the first part I want to talk about is did we use the molecules to overcome mainly heavy metal stress, because that was what I was interested in when I came to UWC. So one of the questions was, can brassica plants dim the light? Because dim is one of the compounds. And when an insect or a herbivore eats the plants, then the right conditions, and then it makes diindolal methane, which is dim. So what we discovered was that when we added diindolal methane onto Brassica nipa's plants, which is canola, we improved the growth through certain biochemical responses. And this is just an illustration showing that the plants looked much taller than the control. And these were the biochemical assays that we performed to show that could be the reason why the plants are performing better. The antioxidant enzymes in that cycle is basically the immune system of plants. That is in layman's terms. So we also wrote an article, a layman's article, where we explained that this could be a possible new signaling molecule in plants. And this is my drawing. That's why Prof. Holgate said that I can draw. So when an animal bites an herbivore, then you get all of this upregulation of also molecules. And when you add them, you then basically mimic that. And that regulation, when it's under normal conditions, is maybe in the green zone, normal. And when you add them, it mimics the herbivore action, and you get better plant growth. And then the next question was, is DIM host plant specific? So only brassica species, like your cabbage and the other brassicas. And then we showed that it is not, because we treated maize plants with diindolal methane, and we also saw improved plant growth. So then the next question was, can we alleviate stress tolerance? And we then we debated which model plant. And so we knew that brassicas make them, and we started the initial work on Brassica napus canola. And so it was just an easy transition to use that. And so we treated plants with vanadium stress, and we found that one genotype performs better than the other genotype when we have vanadium. So now we have a model, because now we can study our molecule on the one that performs worst. I hope that makes sense. So this is just showing that AV Garnet is a genotype, and Agamex is also another genotype, and Garnet was the sensitive genotype, and Agamex was our tolerant genotype. And so when we treated the plants now with diindolylmethane, performed better, and that paper was published. And that took us 6 years to publish that paper since the initial work on diindolylmethane. And we saw that we can improve under vanadium stress, we improve chlorophyll content, and if you did biology, you would know that chlorophyll is giving us the green, and it's very important in plants. And we saw that diindolylmethane does not change the content of vanadium, so it must balance out something else in the plant. And we digged further, and we saw that there's an improvement in iron content. And iron is very important also for plants. And the next molecule is methyl glyoxal. So, and this work took us 7 years to publish. So yeah, I've been in the trenches for long. And recently I presented the work. They wanted me to present 7 years worth of work in 10 minutes. So when we studied the vanadium work, we saw an increase in methyl glyoxal, and we then looked in other articles, and we saw under lead there was also an increase in methyl glyoxal. This is inside the plants when they were treated with the metals. I'm not going to go into details about how methyl glyoxal is made. There's 2 cycles, one a non-enzymatic cycle under normal conditions, and then there's an enzymatic cycle. And again, the methyl glyoxal synthase is not known, which is very interesting. But there are other enzymes which they have identified which can make methyl glyoxal in plants. Methyl glyoxal also shares a link with diabetes. So that was our link also back to the plant-human health nexus, because the next step would be to use diindolylmethane and methyl glyoxal to target some of these diseases. And again, when we treated the plants, this time with a metal called zirconium, we saw that there was a decrease in the plant dry weight as well as growth. But when we added methyl glyoxal, we saw that we could recover that effect. And again, the antioxidant defense was the key to that whole cycle. And this is something that takes me back to my PhD years, because we studied this in detail. And then also cell death. So when we treated plants with zirconium, there's an increase in cell death. And when we treated with methyl glyoxal, there was a decrease. And in this case, we noticed that the metal translocation was affected by methyl glyoxal, not like in the case with DIM, where there was no change in vanadium content. And then again, I draw this very beautiful diagram, and our referees or the reviewers were very happy to see this, that somebody out there can draw such beautiful graphs. Okay, so to our pharmacist, you can actually close your doors, and we can actually now open the stall in the pharmacy building, because the plants give us everything which the pharmacist will give you. Maybe not in the right concentration, but yeah, that is debatable. Okay, so in the next few slides, I'm going to show how we used the fungi that's associated with the plants and how we use that to study the plant-human health nexus. So the first study was where we looked at a disease-causing fungus, Fusarium oxysporum, and we then used a plant endophytic fungus, Boviana bassiana, and we know that this fungus controls insects, and so they use it as a pesticide spray. But no one knew what it can do under other infections. And so this work was done with our colleague at CPUT. And so this image was prepared by Dr. Gerard Basson, who came on board, and because the reviewers were back and forth or debating with Prof. Nchu about whether his fungus is Fusarium oxysporum, and Gerard Basson showed that with his techniques that it is Fusarium oxysporum, and it was the tomato one, Lycopersicki. And yeah, the people at CPUT, they do things reverse. So this is the control, this is infection, this is the endophyte, and this is the combination. And you can see the combination even looks better than the control. So there's something definitely going on there. And here you can clearly see the plant height. This is more than the control, and the number of leaves also, just slightly more leaves in the combination. And so one of my students, Cedrina, she got samples from our collaborator, Professor Vuyo Mavumengwana, and we isolated various fungi. And we treated common bean plants, and you can see that the whole plant length was increased by this one here, and she named it Siliu. And also, when we looked at the pod number, because we are interested in giving people food, and pods in common bean are the essential part. And we saw that when we treated with this fungus, we get much more pods than in the controls. We saw the same effect in the chlorophyll, and this time it was under vanadium stress. So when there was vanadium stress even, she improved the chlorophyll content. Then I read up further. There's now a drive to actually extract chlorophyll and then to use it now as supplements, as well as for beauty and skincare. So green stuff all on your face. And apparently it's working. So that was something interesting. So we can treat the plants with this fungus and then harvest the chlorophyll, and then we can use it as these products. And I'm looking forward to having some guinea pigs to come forward, to come and test it. What we saw under the treatment again was that in the leaf, there was a decrease in vanadium content, but there was an increase in vanadium content in the roots, which shows us, or to us, that this fungus blocks the translocation of vanadium from roots to leaves, which is also, again, very interesting because that will lower the vanadium in the pots. So we will have safer food to eat. Then we also went to the False Bay region, and we got some fungus from there, and we then tested the fungus on a model that's very similar to Mycobacterium tuberculosis, M. smegmatis, and we showed that one of them could, when we take the extracts, it can block the growth of this bacterium. But we took it one step further, We tested it also on Mycobacterium tuberculosis, and we saw that, again, the same fungal extract can block that growth. So hopefully in the near future, there can be a treatment coming from the work. And this was from the European sea squid. And so, very interesting. We also identified some metabolites, and one of the metabolites that stood out for us was bionectin. And when we did the molecular docking, where we now use computers to now see if bionectin can bind to certain of the proteins inside these bacterium, we saw that bionectin strongly binds some of these proteins. And so I'm not going to read all of the conclusions, but it was very, very interesting. And it's showing us that these fungi from the environment or from plants can be used to not only give us more food, but also give us drugs in order to fight diseases. I'm not done yet. So what did we do in cancer? We then just walked around on UWC and saw this strange-looking weed, Coniza bonariensis, and we knew that it's a very hardy weed. It survives. And so we, again, extracted the fungi from there, and we found an alternaria species in the root. We called it DR13. We again extracted the metabolites, found some metabolites that were very interesting, and then we did an assay, a resazurin assay, which starts off as blue, and if there's viable cells, it turns the plate or the liquid pink, and if you block that, it remains blue. So if it's viable, it turns pink. If you block that proliferation, it remains blue. And we did the work on a breast cancer cell line, and we saw that this fungi here, DR1, at lower concentrations, actually cause more proliferation of the cancer line. And when we used DR13, we blocked it at all of the levels. So this goes two ways. If you get into contact with DR1 and you have a predisposition or anything breast cancer alike, you might be in trouble because that might lead to more proliferation of the cancer. Also DR7. But then, luckily, we have DR13 for you to save your life if that happens in the near future. So again, when I saw this, I was jumping up and down, and then my collaborator, Prof. Vuyo, had to calm me down and said, just relax. Just relax. So I would, at this point, like to thank the 40 honor students, 40 MSc, some of them overlapping, the PhD students, postdocs, national collaborators. If I left one out, please forgive me. And then the international collaborators who's probably watching online. So where are we going now? What is the future of plant biotechnology? And this is probably going to be me when I walk out of UWC. So I foresee artificial intelligence playing a big role. Synthetic biology is going to come in. We're going to make new bacteria. We're going to make new fungi, hopefully. Maybe we can put up a new plant. Nanotechnology is going to feature. Big data science. That is the 4 pillars that I see that's going to come in. So what have we been doing? 2 gentlemen, Dr. Sam McInturf and Dr. Kay Gao, they came over to UWC and they were tasked to build a phenotyping robot in 2 weeks. And we all knew that was crazy. But they thought they can do it. A lot of bulbs were blown in the process. And I would just like to come to this. it was also stated by Prof. Galagala in his talk the other day, AI is not going to replace humans, but humans with AI are going to replace humans without AI. I agree with Prof. Galagala. So, yeah, a lot of bulbs were blown, and eventually, after 2 weeks, there is our rector, smiling, because we were told this thing must move at least. The camera here must move. And so when Kay started up the computer, it moved. Everybody was very happy. This is Prof. Moon Choi who is the president from University of Missouri. So this is the camera, and you can put your seats like on here, and then the camera will move, and it will take pictures. And this is what we cannot do, because you cannot sit there day in and day out to look at these seeds germinating and growing. So this is an experiment that we set up. We know that the cultivar Ivy Garnet germinated poorly from our previous study, and we then treated it with various bacteria, and we found that the 1-pentoeagglomerates R6 actually decreased the germination rate. And it also makes the plant now perform even poorer, much poorer than the control, because this is the control. And this is over 14 days. But I'm showing the video in a minute. So this is the power of AI as we are looking at the screen. So this one here, Y1, is a bacteria from my postdoc at the time, and it looks like it actually improves the growth of Brassica napus. And then my postdoc left UWC. So we never took that work further, but we carried on the work with pentoeagglomerates R6. This was the work of my PhD student, Leanne Niekerk, and she also treated the plants with I3CA and then used the robot now to track what is happening under iron deficiency stress. And this is a collaboration with my collaborator, Professor David Mendoza Cozatal at Missouri. And what Leanne found was that when she's got R3CA and iron deficiency, the plant looks like the normal control. And that was the end, actually, of her PhD. She graduated. But yeah, we also looked at proteomics analysis. This is the global proteins. And we found 3 important proteins: a glutathione S transferase, a cysteine protease, which took me back to my PhD days, and an oxalate CoA ligase. And we are looking forward to investigating further what is the role of these proteins in plant growth. Then also, I said nanotechnology is going to come in, and my MSc student, Zaira Omar, then bought quantum dots. In our chemistry department, we also have a professor who's an expert in quantum dots, and she treated common bean plants and showed that it actually moves from roots right up into the pots. And you can see the yellowing there. So it looks like it's inside, actually in the seeds, which is scary. And now we can use that and combine that 2 metals, and then we can track how the metals are moving the nutrients in the plants. And we use the system, the IVUS Lumina. Then we also looked at the bacterium. We isolated the bacterium, and we did all of the characterization on the bacterium. And we then treated Brassica napus and treated it with antimony, which is also a metal, a toxic metal. And when we treated it with antimony, all the nutrients went down. But when we added Bacillus firmus, we brought back all of those nutrients either to the control or over the level of the control, which now means that, again, we have a bacterium that can regulate nutrients in the presence of these toxic heavy metals. And we combined that expertise with nanotech. So we used the quantum dots again, and we put the quantum dots this time inside the bacterium. And so this is not straight onto the plants. This is inside the bacterium. And then we treated the plants, and then it allowed us to track the movement of the bacterium inside the plants from roots to leaves. And this idea I took from my collaborator, Professor Mendoza Cozatel, who always in his presentation shows this. He doesn't use a Golf, he uses another car. But the importance of the picture is, without the parts, you cannot make the car drive, right? And it's going to be similar to synthetic biology. We will have to use all of our knowledge on nucleotides, on proteins, on fatty acids, all of that, to build an organism from scratch. So where do I see, which organism do I see, which plant? I think it's going to be something similar to Macanthia polymorpha, which my last visit to Missouri, I worked on this plant. On the time scale, you can see that it's one of the oldest plants. It's 450 million years old. And David told me, you are now in the presence of greatness. And I think he meant greatness, because it's extremely old. And it is very easy growing. It doesn't have the complicated parts like the flowering plants. So I'm foreseeing that it's going to be something similar to this that we will build from scratch. We already did that, not us, but other researchers with bacteria. They managed to build a genome from scratch and then put it in a cell, and they showed that it works. So we started working on this now in my lab, control, and we treated it with cadmium. And we can clearly see that cadmium does have an effect. David does not believe this. He says Macanthia polymorpha is much more resistant. In his lab, it doesn't show this effect, apparently. I hope by now it does have the effect. And when we treated it with metaglaoxyl, you can see it looks, again, similar to the control. So we have a model, we have bits and pieces, we just have to now stitch all of them together. And it's clear also that there's much more rhizoids. This plant doesn't have roots, so rhizoids. It does have much more than the control even. And then with the bacteria, we sequenced the Pentoia agglomerans, which was looked like a pathogen in the Brassica napus work. And my student Darren treated the Brassica with a mutagen, and this is just showing how the mutations take place. And you can see you start off with a GC and you end with AT. So those changes will all lead to changes at other levels, like proteins. And this will have a clear impact on how the bacteria will grow. And we saw that when Darren is now doing time lapses of the bacterium. One of his mutants are growing much more bigger. I don't want to use the word, but it looks much more, grows rapid than the control. And the other one looks very similar to the control. So when Darren treated the canola again, and we know from our previous results that R6 have an effect on germination, Darren found two mutants, W10 and W16, which is actually these two. And he found that they improve now the growth. So Darren basically changed a pathogen, which is bad, to something that can potentially be good. And now we can study how does this work and why. So what makes R6 a pathogen? And we can now use the tool as a model. But also, my other student, Amira, took this to Stellenbosch, and she then tested them on the breast cancer line. And all of them shows very nice results. And this is the control, cisplatin, which is a drug used in chemotherapy. And W16 at lower levels was our best performer, even outperforming the cisplatin. So this is preliminary results, but we are looking forward to what is going to come next. We also tested it on vero lines, which is a kidney cell line, but from a monkey. And we show that there is no effect with all three our bacteria on the cell line. And it is known that cisplatin does have an effect on this cell line. The thinking behind this is that all of our bacteria and fungi will have to be put through a test like this to make sure that we don't have a bacteria which we're going to take to the fields and that is going to be negative on the cells. So, but Amira wants to run away now to Stellenbosch. So, hopefully we can still get her to do the test. This is all my new students, and I must say thank you to all of them for all of the work that I showed now in the future. And then the funders, the Center of Excellence have been with me for 10 years, and also the South African Education Program Symposium, AMSEP, also Grain SA. And this is I want to say thank you to my whole department. This is not farewell, like I said. And all the past members that was also there with me. Some of them resigned, some of them retired. And then my firm foundation, my wife, Jessica, was also my personal chef, and also my fashion, she dresses me. The two, we are patriots, as you can see. And then I want to leave the students with us. This is me. I was a beautiful boy. And so the future belongs to those who believe in the beauty of their dreams. At that stage, I never would have dreamt that one day I would stand here to present this talk to you people. And some of you who have been on my journey from the start are here today. And I want to say thank you to the faculty who supported me from day one, and also all of you that came out here tonight to come and support me. Thank you.
I think congratulations once again, Prof. Keyster, on your journey and your scholarship. Let's give him another round of applause. I now call upon Professor Ashwell Klein to respond to the inaugural lecture.
Good evening, everyone. So I have the most difficult task of trying to squeeze in a response of Marshall's 100-slide lecture in less than 5 minutes. I will try my best. Please bear with me. But I think before I start, I had it all mapped out here. I wrote it down. And I think when Marshall started talking, I think all of this flew out of the window. So I don't know what I'm going to do now. I honestly don't know where to start. But while I was sitting there and gone through this very deep reflection of what Marshall was presenting today, I realized that, there is a common denominator in everything that he's been presenting there. And that's actually me. I don't know if all of you have paid attention, but in most of those things that you've highlighted, I'm there. So if I can really respond to Marshall's lecture, and I'm sure everybody would agree with me, it should be Marshall plus microbes plus plants equals biotechnology, or the SDGs 2 and 3. And I think that in itself sums up Marshall's entire 12-year career at UWC. He's a very passionate person about what he does, and he's got this never-die attitude, and I am just super grateful that I could have walked this path with him. But then I was told, you only have 5 minutes to speak about this talk of Marshall, and I was like, I need an hour to speak about Marshall before I can speak 2 minutes about his actual inaugural lecture. But I'm probably not going to go into that. The one thing that he left out, though, and you would know, is that he didn't make mention of Dragon Ball Z. In all of this, Dragon Ball Z was probably a high priority while he navigated his way through his entire academic career. So I think with all that said, I would like to take this opportunity on behalf of the senior management, faculty management, and a very proud biotechnology department to say, Marshall, thank you. Thank you for delivering such an insightful, thought-provoking inaugural lecture. Your work on the interconnectedness between plants and human health and its implications in global food security and the overall well-being of us as humans, it provides us with a comprehensive understanding of the important role that plants, microbes play in sustaining human health. So I will try my best, like I said, I only have a couple of seconds left, to summarize or at least extract some key elements from his presentation. But before I do that, I would like to start with a quote by Carl Sagan. And I think, again, that quote holistically sums up what Marshall was presenting today. And it goes as follows. You need to know your past to understand your present in order to predict your future. And his lecture today has taken us through that particular journey, where he came from, where he's currently at in his academic life and personal life for that matter, and where he sees all of this going. And for that, I salute you, my friend. I salute you for that. So, Marshall, you took us on your journey, and you gave us some insights into where it all started, and where you currently are, and where you are heading towards. But for me, what really stands out is that you start off by remembering those that have walked in front of you, that have essentially paved the way for you, and you are acknowledging those that are still walking with you, and that's your family. Me, being a family man myself, we value the existence of our family. They are the cornerstone. They are who we are. They define who we are, and I have seen that in Marshall, and maybe that's why I have such a very strong gravity towards him. And maybe during the break time, if you really want to hear some stories about myself and Marshall, you can speak to him when we have maybe some refreshments, and he will tell you what we talked about when we came to UWC. We started together, and we made a pact that we are going to chase the original idea. Clearly, we are still chasing. We haven't found it yet, but based on all the innovative stuff that was presented today, I think, Marshall, you might have found a few, but I know this is not a sprint, it is a marathon, and the marathon will continue. So something else I would like to also point out or highlight is that you also gave us some insight into your early career as an MSc scholar and doctoral scholar, where you focused predominantly at microbes in the beginning, and then later on with plants. And I believe you made a very good choice to stick to plants. And I mean, there's a whole lot of reasons for that in particular, since you are the chair of sustainable agriculture at UWC. So it appears as if your love for plants really followed you in your academic career, where you initially started working with signaling molecules under the supervision of Professor Ludidi, and you also used that as your initial benchmark or start-off point for your academic career. If I can point out two projects that he actually highlighted, which I thought he was not going to mention, is a cumulative 7 to 9 years worth of work where he focused primarily on DIM as well as on methyl glyoxal. So he started off working on nitric oxide and CGMP, but then his team obviously with him at the helm, they started working on DIM, which is a novel signaling molecule at the time, and they have shown not only can this molecule improve plant growth under normal conditions, but it can also help the plant activate its natural defense mechanism to cope under environmental challenges. And he did exactly the same thing with metaglycoxal, something that he initially thought wasn't possible, but with his kind of attitude, he literally said days and nights. He would even call me sometimes at night saying that, I think we should follow this suggestion. And knowing Marshall, irrespective of what I will say, he will still continue with it. So I just end up agreeing with all he has to say, and it kind of paid off based on those two high-quality impact publications, and especially the metaglycoxal that was widely publicized. I mean, there was a lot of news articles associated with that manuscript. So I'm just glad to be part of that particular journey. And for those of you that do know Marshall, and I think if you want to really know who Marshall is as a person, because in his own opinion, he is a self-proclaimed introvert, I would say speak to his family and speak to his students. That might be a completely different view. He is who he is, and he's a people's person. Whether he likes it or not, Marshall, you are not an introvert, my friend. I mean, this 200-slide presentation was a true testament of that. You are not an introvert. Again, for those of you that have followed the presentation or his career journey like I have, would know that sustainable crop production and food security is very dear to him. And that's why it's no surprise to me that he chases the SDG 2, which is zero hunger, as well as the good health and well-being as part of his research vision going forward. I really would like to delve deep into this interdisciplinary kind of research, but I think you already told us extensively what that is about, so I'm not really going to go too much into that. The fact that Marshall is a biotechnologist. He doesn't shy away from any challenge. It doesn't matter how big or how small it is. So him being a plant biologist by training, or a biotechnologist by training, anybody from anywhere can come to him with any idea, even if it's from the arts. You will still pursue that challenge. And chances are he will put 200% in it because he believes in what you believe. And if you can sell your idea to him, you have his full backing. I know that for a fact. So, Marshall, I also would like to mention that you've placed a lot of emphasis on interdisciplinary collaborations, which is the way forward, especially if we want to start building a resilient environment, a resilient community. We can no longer be working in silos, and I think the stuff that you presented today is a true testament of your willingness to engage with others for the greater good. So I only have like a couple of seconds left. Please bear with me. So the last part of your lecture talks about the future of plant biotechnology. And again, you had no choice but to talk about it, given that you are the chair in sustainable agriculture. But in your lecture, when you mention the future of plant biotechnology, you highlight specifically the importance of collaborations across different fields and institutions, the collective efforts, but what is more important, and I think you did it at your last slide, is you acknowledge the contribution of your peers as well as the students, because without them, this 12 years worth of work would have been a blank slide. It would have been a blank presentation. So thank you for that. And I hope for the students that are currently here, you have a very good supervisor in Marshall. You have a very good leader in Marshall. Sometimes a bit stubborn, but well, that is for another day. But I think just for me to wrap this up, because I don't want to keep you any longer, is that Marshall, your inaugural lecture was not only a celebration of your past achievements, but also inspired a sense of responsibility towards advancing research and practices that will contribute to global food security and human health. So we all are looking forward to seeing how your work will continue to contribute to the greater good of food security and human well-being, and we thank you for sharing your expertise and your futuristic vision with us. Congratulations, full Professor Marshall Keyster.
Thank you, Professor Klein, for your response. I now call upon Professor Rian Denhan, who is the chairperson of the Department of Biotechnology, to give the vote of thanks.
Thank you, Prof. Duba. Oh, Ashwell, we're not compatible heights with this thing. Right, good evening, ladies and gentlemen, from my side as well. I realize I'm standing between you and supper, so I'll keep this brief. But we'd be remiss not to thank a few important parties in this. As you can imagine, an evening like this doesn't just happen. First of all, I'd like to thank members of the senior executive for being here, especially Professor Pretorius. Also, the deans attending, including our new Dean of Natural Sciences, Professor Holgate. I'd like to thank Professor Admai Duba also for what he's done for the faculty in the last few months, taking care of us, and as well as being our program director this evening. To our respondent, Prof. Klein, thank you for that fantastic summary. It's clear that your relationship with Marshall is improved by your personal relationship, your friendship. And it's a joy to see you guys interacting in the lab and collaborating in this way. I think it's to both of your great advantage. I would like, just in terms of making the evening happen, thank you to the Institutional Advancement for arranging the musical items and the soloist, Ms. Tempe, for leading the national anthem. From the faculty for their arrangements, Tasneem Parker and Khadija Parker, thank you for all of your arrangements. And then members of the academic procession, including Prof. Holgate, who managed to not quite fall by the end. And then all of the guests here from the various departments in the faculty, from outside the faculty, everybody who knows Marshall personally, thank you for being here and making this a very special evening. And then finally, just for myself, for Marshall as well, when the call came out to nominate people for this inaugural lecture, I quickly asked Marshall, I said, you'd do that, right? And he said yes, without really thinking about it. And because I hate unanswered emails in my inbox, I just send the reply immediately, yeah, Prof. Marshall will do it. And I guess we were first to answer, so you didn't look like you would thank me for it yesterday, but I think now we're all very happy that we're here, and thank you very much for your thought-provoking lecture and giving us insight into what you are doing and what you are going to continue to do to take the subject forward in the country, and with all your fantastic visions for how we can improve the world with biotechnology. And then finally, I was handed this and told it's not mine, so if you can just come forward and accept this, please.
Okay, thank you, Prof. Denhan. And we're also arriving at a time of taking photographs as well, so there'll be lots of opportunities to take a photo with Professor Keyster. So with that being said, I would also just want to thank everyone who has come out tonight. And now we are at the end of this event. So we will have refreshments. They're being served upstairs in the foyer area. So what I will ask is, we've got an academic procession, so I'll just ask us to all stand. And then the academic procession will proceed to go upstairs. Then we can all follow to go upstairs as well. And then please, let's take as many photos as possible together with the family and with Prof. Keyster. And thank you once again. So we will all rise. Thank you.